Okay, I've started the recording. Here you go. After you, Niraj.
Hello, everybody. So I'm Niraj. I'm a second-year master's student in the Department of Computer Science, and I'm presenting today on behalf of my lab on the work we do for developing deep learning methods for segmentation and analysis of cryo-electron tomography data. So our lab is located in East Campus, JSC BU, and to know more details about our lab, you can also hit our website, cryo-electron.colorado.edu. And so the agenda, I'll be talking about our team, what we do, how it helps in segmentation of bioimages and analysis, and also our current projects, the challenges we face, and the methods we use, and why we also use them. So let's get started. So our team, this is a coordinated effort between our lab and the LBNL lab at UC Berkeley. So from our lab, I report to Assistant Professor VIgnesh. So our lab is again located in JCBB, which is right on East Campus. And from LBNL, we have staff scientist Peter Schwartz and also a postdoc research Eric Roberts. So their team is basically called CAMERA, the Center for Advanced Mathematics and Applied Research. So the team is basically of applied mathematicians, computer scientists who develop mathematical ideas. And it is also supported by US Department of Energy. So they also develop tools and other machine learning libraries for bioimages, especially. And our lab basically focuses on visualizing and developing methods for analysis of cryo ET data and ET data. Apart from that, we also have other biochemistry projects, which for those details, you can also check out our lab site. So most of our work revolves around 3D electron tomography data. So electron tomography data is a technique which provides three-dimensional information or a view of the cells and tissues inside it. So if you look at the video, we can see the membrane, all the other cytoplasms, RNA, etc. And this has been remarkably growing across last decade, and there has been a lot of scope in this field. Although there has been a lot of data for this, but there's not enough automation to analyze this data. So think of it like you have a 3D volume of this data and you want to extract one part of it, suppose DNA. So you would have to label all of them. And then, so the 3D volume would have like 600 to 700 images. Imagine labeling one of them, every one of them, and then visualizing it. That's labor intensive and that would take too much time. So that's where everybody's been pushing towards developing automation in machine learning and deep learning to segment this automatically or generate 3D visualization of this. So although there have been development in creating workflows for this, but most of that requires a lot of user interaction, and not everybody is familiar with deep learning or computer vision. So if you present a lot of code or deep learning frameworks to a biologist, you'd have a hard time going through each one of them. So our goal is to create automated workflow where you just put an image and you click a few things, and then it could show the visualization or provide some analysis with minimal user interaction. And apart from this, not everybody has good compute power, so we have to do this with restricted amount of memory or CPU. So the challenges we face is, first one is the pixel by pixel segmentation of the full image or the volume. So we have a 3D volume and which has like 600 to 700 images, and we have to segment each pixel, and it's not just single or two classes. There's like 4 to 5 classes. How do we segment it? And also any deep learning framework would require you to annotate that. So we have to annotate 3D volumes, or can we do it in 2D? So that's one of the questions. Also, usually deep learning would require millions of parameters, and not everybody has a high compute cluster. So what if you just have a small lab system? Can you train your network there? And can you segment all the images and analyze on that without having to pay extra for the cloud or other performance cluster? And the other problem is the shallow contrast profile. Now, most of the work around this has been outside the nucleus. So the right side image shows the structures which are of high contrast. So when we draw a contrast profile across that line, you see the red ones are the one where most of the work has been done, where the contrast is pretty good. But the left one is internal structures of the nucleus, which we are trying to segment. If you see the contrast profile, and it's very low. So the signal to noise ratio is like very small, and the network usually gets confused between, is this the background or is this a feature? So if you closely look at the image, the feature and the background are very similar. So that's one of the challenges. And apart from this, user experience as well. Nobody wants to, go look at the code, debug it, install it, and spend the time changing the parameters, etc. So how do we accommodate all this constraint into a single workflow? That's why. So this is one of the sample data which we have. So the right side is a 3D volume of Cryo-Electron Tomography. So if you look at it, it just looks like some black dots, and we have to segment this out. So the left side is one slice of it. So if you combine 600 of them, the right side image would look like that. So that's the whole 3D volume. And we want to extract the structures, the lines, which are called the filament, and the bigger ones is called membrane. So we just want to extract those, and they look very similar to the one in the background. So directly starting off with this data, we thought of taking first a simple data, so this is called electrotomography data. So this data has a better contrast, higher resolution, as well as it's a smaller in size. So the whole idea was to have a general workflow. If that general workflow works for this, then fine-tuning that workflow, maybe add some pre-processing, which should work for cryo-electron tomography. So for this data, we have 3 stacks, like RI1, 2, and 3. You can assume that 1, 2, and 3, each of them are like samples from a huge data. So if you say you have 100% of data, first 10%, middle 10%, and the last 10%, in simple words. And each of these stacks, it's like you have like thousands of images. So overall size is around gigabytes. So this becomes a big data problem. Now you cannot annotate everything or segment all at once. So we have to develop workflows where we can use smaller chunks of data, do batch processing, and segment all of it at once. So the data looks something like this. So this is an XY view. And when you go down in Z, a nuclei, so let's take one of the nuclei in the middle. So it grows and shrinks. So that's like one complete nuclei. So we want to extract the complete nuclei and identify the structures inside them, segment it out, and visualize in 3D. So this helps them understand, okay, how the internals of the nucleus are changing, which has other applications. So visualizing the raw data, all of them together, is impossible. So this will be really helpful for that. So shrinking and expanding is one complete nuclei. So like this, we have like 15 of them. So each stack would have like 15 nuclei. So we have like 15, 15, 15, around some 45 nuclei. So for the workflow, we use deep learning. In short, deep learning, you could say, is a functional approximation of input to output. So you pass the input, you have some function, it gives you some output based on that. And for deep learning, there are a lot of networks. And for this particular use case, we use two different networks. So we have a UNet and a MSTNet. So UNet has been popular for biomedical segmentation for a long time. It's because it's very robust, it's simple, and it passes the contextual information very easily. But the problem with units is that it requires a lot of parameters. So a typical unit which we use requires like 9 million parameters. So you have like a big image, a big network, you combine them both, you have to fit in a 24 GB GPU. Sometimes it's not possible. Even if it's possible, the amount of training required to do that would take like few hours or more. And the other problem would be, suppose you're not able to annotate all of it, and you annotate like 30, 50 of them. With millions of parameters and low training data, your model would overfit, and that would not generalize well. So those are some problems with UNet. But if you have a good amount of training data and you're able to annotate all of them, like most of them, then it would definitely perform better. So that's where mixed scale dense network helps. So this is really useful when you have like very less training data. So with low training data, it's actually like 2 to 3 times less parameters compared to units. So this will be really helpful when you have smaller systems. In terms of accuracy, which we have seen, both perform really well for data. So it just depends on how much resources you have and the use case which you are trying to use this for. So we have the network. Now we need to extract nuclei from the data sets which we have. So the major problem in this is, so the first thought that would come on our mind to extract a nuclei is, since the nuclei is almost spherical in shape, we could just use some contour detection, detect the boundaries or the edges, and just extract it out. But the problem with that is, although the membrane appears to be continuous, there are gaps in that. So when you actually do the contour detection, it detects every piece as a different entity. So it also requires some kind of pre-processing. The other problem is when you scroll the image, like go over the image from top to bottom, the contrast varies. That's just how the data is generated. So this is the contrast profile of the data. As you see, so from the left is the topmost part of the image and the right is the bottommost. So the contrast gradually decreases. So it's not uniform across the image. So that's why we use adaptive threshold, so that we take a box around every pixel and it would just match the threshold. And then we also need to fill the gaps. And for that we use morphological gradients, which uses a combination of dilation erosion, which is nothing but you remove few pixels, add few pixels, just a combination of that to fill the gaps. And once we fill the gaps, we can just get the contours of it. We know nucleus are spherical in shape, and the biggest boundary in the image would be the nucleus. And we generate mask and we cluster all the nuclei, so that this would in the end have like 15 folders, where each folder would have nuclei images. And in the end, we would have one single nuclei for all the 80 gigabytes to 200 gigabytes images. So once we have the raw data all pre-processed, the next step would be to annotate it. So we are basically interested in 4 different classes of the nuclei. So the yellow is the nucleolus, the green is the synaptic complex, which we usually call SC in short. The brown is the chromosome, and the blue is the membrane. To improve the network performance, we also label the rest of the nuclei with some other class. This actually increased our accuracy by a lot, so that the network doesn't get confused and simple. So this is how we annotate the data, and we just annotate like 1% of the data out of what we have. So this makes it really easy for the user to adapt. So for the training, we have a single GPU in the lab with 24 GB memory. We have a unit which works best with 9 million parameters were tested. And so to verify if our network is behaving the way it's supposed to, we use 10% of the data for validation and 10% for the testing. In case it starts to behave unexpectedly, we could do an early stop that would save us some time and memory. Once we have the results, so based on what we have, we use the F1 score, so which is nothing but the combination of precision and recall. So precision would tell us out of actual positive, suppose we have chromosomes, out of all the chromosomes, how many did we actually mark as chromosomes? And recall would tell out of the total data, how many chromosomes did we actually validate. Just having one of those would not be accurate. So we use F1 score to correctly classify all the classes. So this will give you a good metric to evaluate all the different classes which we want to predict. And since the number of pixels in the nucleolus or the chromosomes might be different, so one might have more pixels and one might have less. So we use micro F1, which is like a weighted average of all the scores. So that would give importance to all of the classes equally, so that the predictions are. So the tech stack we used for all of our training and segmenting. So the first one, we use a PyMSD torch library. So this is developed by UC Berkeley LBNL team. So this allows us to create flexible and custom networks so that we could modify the network for every use case, because not every network would work the same for different cryo data. Some might have low contrast profile, which would require more parameters. So you could just look at the documentation and just change a few variables. You can easily create your own custom networks. You don't even have to, deep learning in depth for this. Second we use is the Quality 3D. So this allows us to slice the images. Now, suppose we have developed workflows for 24 gigabytes of memory, and your lab supports only 12 GB of memory. Now you cannot change the unit parameters because that is tested, but what you can do is you can slice the images. So this allows us to slice those PyTorch tensors. So you have like 1200 cross 1200 images. You can pass like 256 cross 256 multiple images to the network. Although your processing would be a little slower than expected, but it would fit into your GPU size. Now, this is not same as resizing, because resizing would change your resolution. This would just slice your image, so your resolution remains the same. And then for visualization, we use Napari and rest of the common libraries which we use for batch processing and faster parallel processing. So our overall workflow looks something like this. So we have like TIFF stacks, all the RI1, 2, 3, we do pre-processing, we extract single nuclei, we annotate them, we train them. So here we annotate only 1% of the data. And we also, while extracting the samples for annotations, we pick few samples from every nuclei so that as you know, the contrast on the upper half of the image is different from the lower half. So if we pick just the upper half of the image, and some of the nuclei are in the bottom half, the network would behave differently. So it's important to train the images on different contrasts. And after training, we segment and visualize the data. So Napari easily has libraries and functions which you can write in Python and Jupyter Notebook so that you can create better visualization, 3D visualization. And those are the images from Napari. So we segment each of the class. We can also view them separately. And the top part is, these are the chromosomes. So we also want to split the chromosomes and see how the structure looks like. So just the segmentation is good for visualization, yeah, it looks better. But what biologists want, they also want to have some metrics to say, okay, so the nucleus look like this in one of the stack. How did it change over time? Did the chromosomes move here? What was the distance from the membrane? Something like that. So these are the results for one of the nuclei which we had. So we have the membrane. So they have the pores, which we are really interested in, and the chromosomes, the SC, and the full nuclei. So these are the internal contents of the nuclei. The green part is the nucleolus. So if you look closely, the chromosomes encloses the SC. And they are not continuous. I mean, they're supposed to be continuous, but there are small gaps. Now, these are because our network didn't do a 100% job. So suppose it did a 90% job, then 10% of the data is missing. And those are the gaps which are shown in the data. So how could we, make it better? So some of the things which we look at after we have the segmentation, how can we remove those false positives and maybe add some more pixels wherever it's missing? For the analysis, so we also want to look at how the single chromosomes are aligned. Now, the problem with splitting the chromosomes is that we have a 3D volume of chromosomes now. So most of the networks, YOLO and whichever are available, for instance, segmentation, do a really good job in the 2D image. So suppose two chromosomes in a 3D volume cross path across each other. So the network will not detect them as a separate chromosomes. So that's one problem with the neural networks. Also, we don't want to add any additional network and have it annotate again the segmented data, which would again you'd spend so much time on annotating, creating a network, again, one more workflow. That's just additional overhead. So the other thing we also look at is the 3D connected components, which will just look at the neighboring pixels from top to bottom. But then the problem is when there are gaps between the 3D volume, how will the output be? So if you look at the right image, the green and the brown ones are supposed to be a single chromosome, but the 3D connected components detects it as a separate chromosome because our network didn't do a 100% job. So if there's a gap, then the C algorithm thinks that, okay, this is separate. So we will label it as a different chromosomes. So can we reconstruct, maybe add the gaps and then run 3D components? So this is one of the current problems which are solving and we're actively working on. And for the analysis, we're looking at, okay, how are the chromosomes aligned? How far are they from the membrane? What's the diameter of the C? What's the diameter of the chromosome of the enclosed C? So something like this. So for this, we would need some complex CV algorithms because we are looking at the 3D volume. And if the structures are crossing path, how do we detect that and incorporate in this? So this is for the first part. So after this, okay, we had the general workflow and we thought, okay, so we have the workflow now, let's run on our main data, the Cryo-Electron Tomography data. So we applied on 2 different data sets and this is what we got. So the left side is the raw image and the middle is the annotations. So which were supposed to, make your network learn, but the predicted output was just predicted everything as that feature. So if you look closely at the image, the difference between the actual feature and the background is like very less. So as I said, so the contrast profile is very low for this image, but for the FIPSM data or the tomography data, it was really different. So the difference in the contrast profile between the background and the actual features was too high. So it worked really well with the ET data. And so annotation for this image is really hard because they are not continuous. So for the FIPSM data, most of the features are really continuous and the resolution is higher. So it's really easy to annotate most of the images. So that's why you could have a last training data for that. But for this, it's not feasible to annotate like 60, 80 images for every data. And then no signal to noise ratio, and the resolution is almost as half as the FIPSM data. So although the networks are same, we could incorporate some pre-processing steps in this that would fix all these problems, and then we could segment this out. So this is our data, which we want to segment. We are interested in the big lines are the membrane and the smaller ones are called the filaments. So we want to extract those out from the 3D volume of the data. So first we annotate it. Now the problem, like I said, in the annotations, they are not continuous. So if you look at the left one zoomed image, so they're like tiny black spots, and in between them, that's just the background noise. So we have to carefully annotate each small tiny part of it so that the network doesn't get confused between the actual feature and the background. So the annotations also takes too much time in this case. So we annotate only like 30 of them so that user doesn't have to spend too much time on annotating this, and we actually incorporate few of the image processing steps in that to overcome the annotation problems. So once we annotate, since we only have 30 images, we use Quality 3D to slice and dice the images. So suppose we have a 1200 cross 1200 images. We split the images into 128 cross 128. So the 30 images now becomes like thousands of images. And we do it at a window of suppose 4 cross 4. So that means it would generate multiple samples of the same image. So that way you have more training data and your resolution is again the same. So overall, if you look at the training process, it doesn't affect much and it really adds value to it. And smaller images provides better performance so that you can also increase your batch size and you can fit it in your smaller systems. So once we fix the annotation and the training data problem, we also have to reduce the noise. So most of the Gaussian blur or any other algorithm, when you apply those noise filters, they also reduce the features. So we don't want to do that. We only want to reduce the noise and also enhance the edges. So we use filters called like bilateral filters, which enhances the edges and reduce the background noise. And we also use histogram equalization to change the contrast so that this would reach similar to the FIPSM data. And then we train the network on these images. And this time the predictions are much better than the last one. So, but this is not the original size of the image. So once we have predictions from all of the smaller images, we combine them using averaging and all the common regions where the images overlap, we average those pixels out, and that is all taken care by the quality library. And so we would reconstruct the final image. And once we segment all the images, so this is how it would look in 3D. So from the black and white 3D volume, we have this. So the left one is from one of our different data sets, which is called Golgi data set, and the middle part of the filaments. And the right one is from the filament data set. So the right one has some of the false positives, which have not cleaned up using post-processing to show the difference. But the left one is after applying the post-processing. So in post-processing, we use scikit images, morphology, objects, libraries, which applies Fourier transform and removes smaller blobs from the images. This would help in cleaning up, but overall, it does like 80% to 85% prediction. So the score is around 0.85, 0.86, which is fairly better than what we have, but we can still improve this by tuning the parameters, try out different networks, and maybe even use MST nets to see, because we can fit larger MST nets as they require less number of parameters. So these are the results from the units. So the current projects which we're working on is first one is the segmentation of chromatin. So till now, what we have done is for the images which have comparatively higher contrast than the one which we are looking at the image here. So this image, the features and the background, like in terms of size, are very similar. So even the edge filters won't work pretty well for this. So we have to come up with different pre-processing techniques to segment this out. And the other one is how do we analyze the segmented data? So right now, the networks, instant segmentation networks don't do a pretty good job. So we have to come up with different sieve algorithms to maybe first fill those gaps and then apply some 3D components. So that would improve the performance of overall segmentation, and then look over how do we find the distance between the chromosomes, where are the chromosomes placed. So it's more of classic CV approaches. So if you're interested in any of these workflows, you can reach out to VIgnesh using this email ID, or you can also check out our lab website. And these are some of the resources that are available. So we recently published a detailed methods paper on this bioarchive. It's the last one. And you can also look at what UC Berkeley and our lab is doing using the following links. Yeah, so that's all I had. Any questions?
Thank you, Niraj. It's very helpful and insightful for the presentation. So if anyone has any questions, please go for it. You can unmute yourself and ask any questions regarding the work, how to connect, the opportunities that's available. So feel free to unmute yourself. I can even stop the recording if that's more comfortable for everybody.